Usually when we talk about a medical diagnosis, there is this underlying expectation of absolute precision.
Right. Yeah, like structural engineering or something.
Exactly.
Yeah.
You break your arm, the x-ray shows that jagged white line right there on the radius, and the doctor just points at the film and says, there it is.
Broken.
Right. It's visible, it's neatly categorized, and honestly, it is deeply comforting to have that level of certainty.
Oh, absolutely. I mean, we really crave that binary, broken or not broken, functioning or failing. It gives us this, well, it's an illusion, really, but an illusion of total comprehension over our biology.
But the moment you step into the world of neurobiology, and specifically stress and behavioral shifts, that metaphorical x-ray machine just completely shatters.
Yeah, it really does.
We stop looking at clean breaks and we start wading into this incredibly murky, constantly shifting landscape of connectivity and neuromodulation. And today, we are wading directly into those diagnostic muddy waters to look at something profound.
I'm very excited for this one.
We are looking at the exact biological moment. Your brain, basically deletes your free will and replaces it with a rigid habit.
It is a fundamental shift in agency. And what's wild is that it's driven entirely by structural remodeling.
Right.
Like, we operate under this persistent illusion that we are the conscious authors of all our actions. But under chronic stress, the biological steering wheel is physically, demonstrably handed over to a completely different set of subcortical neural circuits.
The you that makes careful decisions is quite literally locked out of the control room.
Cut off completely.
And that is the core mission of our deep dive today. If you, the listener, are dealing with a relentless deadline or maybe an ongoing financial crisis or just, the baseline hum of modern existential dread, why do you suddenly fall back on deeply ingrained, often destructive habits?
Why do you snap at your partner?
Right. Or doom scroll for 3 hours or relapse into an old addiction. To answer that, we are tearing into a massive stack of literature today.
A really incredible stack, honestly.
Yeah, we have foundational mapping of the prefrontal cortex. We have these granular electrophysiological recordings from the basal ganglia. And we have a brand new cutting-edge 2025 paper from the journal Nature that finally gives us causal proof of this habit hijack.
It's a chronological scientific journey, really.
It is. Okay, let's unpack this. If the conscious, rational part of the brain is losing control, I imagine we need to start by looking at the specific architecture of that control center before it collapses, right?
Yeah, absolutely. To understand the hijack, we really have to deeply understand the target. And the rational control center is the prefrontal cortex, the PFC.
Okay.
And a brilliant anchor for this is Amy F.T. Arnsten's 2009 review paper. It's called Stress Signaling Pathways That Impair Prefrontal Cortex Structure and Function. It was published in Nature Reviews Neuroscience.
Okay, so going back to 2009 for the foundation.
Right. And Arnsten lays out this really fascinating topography. Because the PFC isn't just, a homogeneous lump of processing power sitting behind your forehead.
Like a single computer chip.
Exactly. It's not that at all. It is a highly specialized, topographically organized network.
So I am picturing it almost like a corporate executive suite where different directors handle very specific portfolios to keep the company, or in this case, you, moving toward long-term goals.
That is actually a really apt framework. So at the top of that executive suite, you have the dorsolateral PFC, the DLPFC.
Okay.
And this region has massive, extensive connections with your sensory and motor cortices. It is your working memory. It's the core regulator of attention, thought, and deliberate action.
So if I'm, say, holding a complex mathematical problem in my head while also deciding which button to press on a machine, my DLPFC is doing the heavy lifting.
Heavily engaged, yes.
So that's the director of operations.
Right. Then, sitting lower down and closer to the midline, you have the ventromedial PFC, the VMPFC.
Okay, what's their job?
Well, its connectivity is entirely different. It basically bypasses the motor and sensory areas and wires directly down into deep subcortical structures like the amygdala.
The fear center.
Exactly, our primary emotion generator. So the VMPFC's job is emotional regulation.
Yeah.
It contextualizes fear and anxiety.
Got it. So that's the director of human resources, basically keeping everyone's panic in check.
I like that, yes. And then there's a third critical player of the suite, the emergency brake.
Ooh, okay. What's the emergency brake?
The right inferior PFC, or RIPFC. This region seems highly specialized for inhibitory control over motor responses.
So like
when you have the sudden overwhelming urge to yell at a client or, throw your phone across the room because it froze.
oh, I've been there.
Right, we all have. The RIPFC is the structure that rapidly fires to suppress that inappropriate physical action.
It's the thing that says, hey, don't do that, you'll get fired.
Precisely.
Okay, so if the PFC relies on this delicate, interconnected balance between operations, emotional regulation, and impulse control, I'm picturing it like a really high-performance engine.
Mm.
It needs premium fuel and exact amounts to keep those three directors communicating.
Yes, and Arnsten actually calls this the delicious cycle.
The delicious cycle. I love that phrasing.
It's great, isn't it? Under normal, optimal conditions, the PFC carefully manages its own chemical environment. It's continuously bathed in specific catecholamines,
Which are what, exactly?
namely noradrenaline projecting up from the locus coeruleus in the brainstem, and dopamine projecting from the ventral tegmental area.
Okay, so dopamine and noradrenaline.
Right. And when you are alert, engaged, but fundamentally calm, like you're focused on a task but not panicking,
Yep.
these chemicals hit the PFC in moderate optimal doses.
Just a steady drip.
Exactly. And they bind to specific receptors that actually strengthen the connectivity of the PFC networks. The neurons fire together more efficiently.
Which means you are capable of deep, flexible, top-down control.
Yes. You're choosing what to focus on based on your internal goals, rather than just instinctively reacting to the loudest noise in the room.
Right, the delicious cycle. But I assume a massive stressor, say, a sudden brutal performance review at work or a near-miss car accident, doesn't just change the fuel in that engine.
No, it completely floods it. It obliterates that delicate balance.
Okay?
Under acute psychological stress, your amygdala detects a severe threat. It immediately triggers stress pathways in the hypothalamus and the brainstem.
The classic fight-or-flight response.
Right. And the result is a neuromodulatory tsunami. Massive, uncontrolled amounts of noradrenaline and dopamine are dumped into the cortex.
Whoa.
And here's the kicker. Because of the specific receptor affinities in the PFC, high levels of these chemicals do not enhance function. They rapidly and catastrophically impair it.
It's the biological equivalent of pouring rocket fuel into a Honda Civic.
Ah, yes.
Like, it doesn't make it go faster, it just melts the engine block.
It melts the engine block.
Yeah.
The very same chemicals that facilitate rational thought in small doses actively dismantle the network connections in high doses.
So the delicious cycle devolves into a vicious cycle.
Exactly. Arnsten's exact words, the vicious cycle.
Okay.
And the architecture of the brain flips. The PFC basically goes offline. You lose that slow, thoughtful, top-down control.
Because the directors are incapacitated?
Right. And simultaneously, those same high levels of catecholamines actually strengthen the operational capacity of the amygdala.
So the fear center gets a massive upgrade while the rational center goes dark.
Yes. Your brain shifts into a reflexive, rapid, bottom-up control state. Your attention is violently captured by the threat.
Okay, now I want to gently push back on this framing for a moment, or at least debate it.
Sure.
Because the term impairment carries a lot of baggage. It makes it sound like stress is just a biological glitch, you know? Like a universal failure of cognition.
I see where you're going.
Right, because anyone who has been in a truly high-stakes emergency knows that sometimes, under intense stress, you achieve a level of hyperfocus and memory retention that feels almost superhuman. It isn't just a breakdown. Sometimes it's a massive enhancement.
Oh, you are touching on a profound evolutionary reality here, and you're completely right. The brain is not just breaking, it is actively reallocating resources.
Okay, tell me more about that.
To really understand that reallocation, we have to look at David M. Diamond's 2007 paper. It's called The Temporal Dynamics Model of Emotional Memory Processing, a synthesis on the neurobiological basis of stress-induced amnesia, flashbulb and traumatic memories, and the Yerkes-Dodson law.
That is quite the title.
It's comprehensive. It was published in Neuroplasticity. And Diamond systematically dismantles this lazy assumption that stress universally destroys cognitive performance.
He goes straight after the Yerkes-Dodson law, doesn't he?
He does.
Which is practically dogma. I mean, anyone who has taken a Psychology 101 seminar has seen that classic inverted U-curve on a chalkboard.
Oh, absolutely.
The basic idea is that, low arousal means low performance because you are bored. Medium arousal hits the peak of the curve, you are motivated, you are in the zone. But high arousal pushes you over the edge, you panic, and performance crashes.
Right, the classic inverted U.
Uh-huh.
But Diamond points out that this simplified monolithic curve is actually a massive misinterpretation of Yerkes and Dodson's original experiments from way back in 1908.
Wait, really? The textbook curve is wrong.
Well, the field reduced a highly nuanced biological observation into a catchy, simple heuristic. If you go back and look, Yerkes and Dodson actually trained mice using electric shocks on two fundamentally different types of tasks.
Okay.
One was a complex visual discrimination task, and the other was a very simple visual discrimination task.
And the results were completely divergent. I remember reading this part.
Yes.
For the difficult, complex task, that inverted U held perfectly true. High stress from a strong shock impaired the mice's ability to figure out the complex puzzle.
Because it requires the PFC.
Right. But for the simple task, the relationship wasn't a curve at all. It was linear. The
Exactly.
higher the shock, the higher the stress, the better and faster the mice performed. High stress strongly enhanced their ability to execute a simple, focused task.
What's fascinating here is how Diamond integrates this with Easterbrook's Q utilization hypothesis
Easterbrook. From when?
from 1959. Easterbrook proposed that arousal fundamentally narrows the range of environmental cues you are capable of processing.
So, tunnel vision.
Essentially, yes. As stress increases, your attentional aperture shrinks down to a pinpoint.
Which actually maps perfectly onto the neurobiology we just discussed with Arnston.
How so?
Well, if you are doing a complex task like writing a software algorithm or navigating a highly nuanced social conflict, you need a wide attentional aperture.
You do?
You need your prefrontal cortex to juggle multiple abstract variables in working memory all at once. So when the chemical tsunami shuts down the PFC, your performance inevitably crashes.
Because you literally drop all the balls you're juggling.
Right. But if the task is simple survival, like focusing exclusively on the fangs of a predator jumping at you or the license plate of a car speeding toward you, you don't need working memory.
You really don't.
You need raw, unfiltered, lightning-fast threat processing.
And that is exactly what the amygdala provides. And Diamond formalizes this brilliantly in his temporal dynamics model.
What does the model show?
He shows that during the immediate onset of a severe stressor, the brain briefly enhances plasticity in the hippocampus and the amygdala.
Enhances it.
Yes. It shifts the hippocampus away from building those complex spatial cognitive maps that it normally does and forces it into a hyper-focused flashbulb memory mode.
Wow. So it takes a high-definition snapshot of the threat.
Exactly. The brain is making a brutal but logical evolutionary calculation here. It's saying, forget about optimizing the long-term strategic plan. Dump 100% of our metabolic resources into remembering the exact sound that Predator just made so we survive the next 5 minutes.
It's incredibly pragmatic.
Very. And crucially, immediately following that brief window of hyperplasticity, the brain actively suppresses any new plasticity
Why?
to lock in and consolidate that traumatic memory. It doesn't want new information overwriting the survival data. So it's an incredibly efficient, albeit costly, survival mechanism.
It is brilliant for surviving a physical attack in the woods, but man, it is a catastrophic mismatch for modern life.
Oh, totally.
Because our predators today are usually a toxic work environment, or an overdrawn bank account, or an email from the IRS. You don't need a flashbulb memory of your boss's angry email.
No, you really don't.
You need your PFC to draft a diplomatic, careful response. But your brain has already cut the power to the executive suite.
Which leaves us with a really profound mechanistic gap in our understanding.
Exactly. This is the transition I was hoping we'd get to. If the PFC, the Director of Operations, HR, and Impulse Control, has evacuated the building and the amygdala is just sitting there blaring the fire alarm and taking high-res photos of the flames, who is driving the car?
Right, because we don't just freeze.
No, we don't just turn into statues under chronic stress. We continue to operate, we move, we speak, we execute complex behaviors. Something else has to step into that void to coordinate our actual physical actions.
And to find out what that is, that requires us to descend deep into the subcortical architecture of the brain. Below the cortex, sort of wrapping around the thalamus, lies the absolute engine room of habit and automated action.
The basal ganglia?
The basal ganglia.
The structural switchyard.
I love that analogy.
If we want to understand how a habit physically wrestles control away from our agency, we have to look at the wiring down here, right?
We do. And the foundational architecture of this whole network is beautifully articulated in a 2010 paper by Andrea Stocco, Christian Labier, and John R. Anderson.
Okay, Stocco 2010.
Yes. The paper is titled Conditional Routing of Information to the Cortex, a model of the basal ganglia's role in cognitive coordination. It was published in Psychological Review.
Conditional routing of information. That sounds like an IT network protocol.
It kind of is. Stocco and his team proposed that the basal ganglia operates fundamentally as a conditional information routing system. It basically sits exactly between the high-level processing of the cortex up above and the low-level execution centers down below.
So a routing system implies it is managing massive amounts of traffic.
Huge amounts.
I want to build out this analogy a bit because the basal ganglia is notoriously complex for people to visualize.
Please do.
Let's imagine the basal ganglia as a gargantuan, highly intricate train switchyard.
Okay, I'm picturing it.
Up above, the cortex is constantly generating thousands of possible trains of thought and potential actions every single second. Like, I should reach for my coffee. I should scratch my ear. I should stand up and walk out of this podcast studio.
Hopefully not that last one.
Huh, no. But all of these potential actions are trains barreling down the tracks toward the switchyard. And the basal ganglia's biological mandate is to decide which one of those thousands of trains actually gets a green light to leave the yard and manifest as a physical movement.
Yes. It is a process of relentless, high-speed selection and suppression.
Okay, so structurally, how does it do that?
Well, the primary input station receiving all those trains from the cortex is called the striatum.
The striatum.
Right. And the striatum is massive and highly heterogeneous. It contains incredibly diverse populations of neurons. Primarily, we're talking about medium spiny projection neurons.
Medium spiny neurons.
Yeah. And they route signals down through deeper nuclei, like the globus pallidus, both external and internal, the subthalamic nucleus, the substantia nigra, and eventually the thalamus.
Okay, that's a lot of structures. But the core mechanism for managing this chaotic influx of cortical train traffic relies on two distinct, opposing neuropathways originating right there in the dorsal striatum, right?
Exactly. This is the mechanism of control. You have the direct pathway and you have the indirect pathway.
And the neurochemical distinction between these two is paramount, isn't it?
It's everything. Let's look at the direct pathway first. This pathway primarily expresses dopamine D1 receptors.
D1 receptors, got it.
When a signal from the cortex comes down and activates these D1 expressing medium spiny neurons, they project directly to the output nuclei of the basal ganglia.
Okay.
And their specific action is to inhibit the basal ganglia's baseline inhibitory output.
Wait, okay, let's untangle that double negative because inhibiting an inhibitor is a classic, frustrating neuroscience quirk.
It really is. It trips up medical students all the time.
So, if the basal ganglia is constantly holding the brakes on the motor cortex like it's naturally suppressing movement, the direct pathway acts by disabling those brakes.
Yes.
It creates disinhibition, which results in a net excitation. So the thalamus is freed up, and it sends a massive go signal back up to the motor cortex.
Precisely. The direct pathway facilitates movement. It selects a specific action from the noise and amplifies it.
So in our train switchyard analogy, the direct pathway is the operator actively throwing the switch to green, accelerating the one single train you actually want to move. It's hitting the gas pedal on reaching for your coffee. Yes, execute this action.
That's exactly it. Now, running parallel to this and constantly opposing it is the indirect pathway.
Okay, the counterbalance.
Right. The neurons here primarily express dopamine D2 receptors.
D2, okay.
And when the cortex activates these D2 neurons, the signal takes a much more circuitous, winding route through intermediate structures, like the external globus pallidus.
But it's a longer loop.
It is. And the ultimate net effect of this longer loop is to drastically increase the inhibitory output of the basal ganglia onto the thalamus.
So it clamps down even harder.
Yes. It is the definitive no-go signal. It actively and powerfully suppresses action.
Back in the switchyard, the indirect pathway operator is basically throwing up red lights across all the other tracks, actively halting the thousands of competing unwanted actions.
No, do not scratch your ear. No, do not stand up during the meeting.
Right, it is standing on the brakes.
In fluid, goal-directed human behavior relies entirely on the dynamic millisecond by millisecond balance between that D1 go pathway and the D2 stop pathway.
Just constantly toggling gas and brake.
Exactly. And under normal conditions, your prefrontal cortex acts as the ultimate wise supervisor of this switchyard. It sends precise top-down signals to bias the selection process, ensuring that the habits and actions given the green light are actually aligned with your long-term goals.
But, and this is the huge but, we have already established that under intense chronic stress, the PFC is utterly impaired.
It's offline.
The executive supervisor literally abandons the switchyard. So what happens to the tracks, the operators, and the traffic lights when the basal ganglia is just left to run itself under a deluge of stress hormones?
It's a great question.
Does the physical structure of the network actually change?
It does. And the scale of the physical remodeling is staggering.
Really?
Oh, yeah. To see exactly how the basal ganglia rewires itself, we need to dive into some state-of-the-art, incredibly granular neurobiology.
I'm ready.
We are looking at a pivotal 2023 paper by Diana Rodriguez and Patricia Montero. It's titled Chronic Stress Promotes Basal Ganglia Disinhibition by Increasing the Excitatory Drive of Direct Pathway Neurons.
Okay, 2023. Published where?
It was published in the Neurobiology of Stress.
Okay, so this paper is an absolute masterclass in cellular interrogation because for decades, studying the striatum was basically like trying to map an overflowing bowl of cooked spaghetti, right?
That is the perfect description. It's incredibly dense.
The D1 go neurons and the D2 stop neurons are physically tangled up together. You can't just look at a slice of brain tissue under a microscope and visually distinguish which is the gas pedal and which is the brake. They look the same.
Anatomically, they're nearly identical.
So how did Rodriguez and Montero actually isolate these specific circuits?
They leveraged highly advanced transgenic mouse lines.
Transgenic mice.
Right. Specifically, they utilized D1 TD tomato mice alongside D2 EGFP mice.
Okay, let's strip away the genetic jargon for a moment for the listeners and for me. TD tomato and EGFP, these are essentially biological highlighter pens, right?
Yes, that's a brilliant way to put it. Fluorescent proteins.
So in the D1 mice, the genome is modified so that every single neuron expressing the D1 receptor, the direct pathway GO neurons, simultaneously expresses a fluorescent red protein called TD tomato.
Correct.
So under a specific wavelength of light, the entire GO pathway just glows neon red.
It lights up like a Christmas tree. And conversely, the D2 EGFP mice are engineered so that the indirect pathway stop neurons express enhanced green fluorescent protein.
EGFP.
Right, they glow a brilliant neon green.
So suddenly the spaghetti bowl is color-coded.
Yeah.
The researchers can take a microscopic slice of the living striatum and see exactly, indisputably, which neurons drive action and which suppress it.
It completely revolutionized how we can study this network. So to understand the impact of stress, they subjected these transgenic mice to a rigorous, chronic, unpredictable stress protocol.
Chronic, unpredictable stress. What does that actually look like for a mouse?
It's crucial because it models the human experience of chronic modern stress much better than a single massive trauma.
Like a shock.
Right. Over several weeks, the mice experienced daily, unpredictable mild stressors. Things like a tilted cage or damp bedding or constantly altered light-dark cycles.
Just mildly terrible things constantly happening.
Exactly. It mirrors the relentless, low-grade grind of a toxic job or long-term financial insecurity.
The kind of stress where you literally never know what the next day will bring, so your baseline cortisol and adrenaline just stay permanently elevated. You never get to relax.
That's the exact physiological state they were inducing. And following this chronic stress protocol, the researchers took acute slices of the dorsomedial striatum,
The DMS.
the DMS, which is a core part of the switchyard. They then performed whole-cell patch clamp recordings directly onto those glowing red and green neurons.
Okay, I have to say, I always find patch clamp recordings to be one of the most viscerally impressive techniques in all of science.
Oh, it requires unbelievably steady hands.
I mean, a researcher is looking through a high-powered microscope using a robotic micromanipulator to lower a glass pipette that is literally thinner than a human hair.
Much thinner.
They press this microscopic glass tube against the delicate membrane of a single living brain cell, apply a tiny bit of suction to break the seal, and they literally record the electrical currents flowing inside that one specific cell.
It is mind-boggling precision.
They are basically wiretapping the switchyard. So what exact metric were they listening for with these pipettes?
They were measuring MEPSCs.
MEPSCs, what does that stand for?
Miniature excitatory postsynaptic currents.
Okay.
Functionally, an MEPSC represents the spontaneous, baseline, excitatory noise or chatter arriving at that specific neuron from upstream regions, primarily a cortex.
So it's the baseline chatter?
Right. The frequency of the MEPSCs tells you how often the cell is being bombarded by a signal, and the amplitude tells you the sheer volume or the electrical strength of each of those signals.
Okay, so they have their pipettes locked onto the red GO neurons and the green stop neurons, and they are comparing mice that have lived in luxury, unstressed control cages against the mice that have been subjected to the chronic, unpredictable stress.
Yes.
What actually happens to the electrical traffic when the PFC is offline?
The findings reveal a dramatic state of hyperexcitability, and it's driven heavily by the remaining deep cortical inputs.
Where are those inputs coming from?
They recorded the projections coming from layer 5 and layer 6 pyramidal neurons within the prelimbic, or PL, and interlimbic, or IL cortices.
QL and IL.
Right. These are the deep output layers of the medial prefrontal cortex that feed directly down into the dorsomedial striatum.
So in our analogy, these are the heavy-duty tracks carrying the trains down into the yard.
Yes, and chronic stress turns those tracks red hot.
Wow.
The increase in spontaneous excitatory drive is immense. In the prelimbic cortex projections, the frequency of MEPSCs jumped from a baseline control average of 1.182 hertz
Okay, 1.182.
up to 2.020 hertz in the chronically stressed animals.
Wait, that is almost a doubling of the baseline signal frequency.
It is, and in the infralimbic projections, the remodeling is even more extreme. The frequency skyrocketed from a control of 0.503 hertz to 1.163 hertz under chronic stress.
I want to pause on these numbers for a second because it is so easy to hear 0.5 and think it is a tiny, insignificant change.
It sounds small.
Right. But in neurobiology, translating a jump from 0.5 to over 1.1 cents hertz into physical reality means the background excitatory firing rate has more than doubled.
Exactly.
If this were a microphone, the quiet background static just turned into a deafening roar. The basal ganglia is being relentlessly bombarded.
Bombarded is the right word.
And what about the amplitude, the volume of each of those signals?
The amplitude increased significantly as well.
Yeah.
Looking again at the infralimbic cortex, the control amplitude was 10.043 picoamps.
Okay.
In the chronically stressed mice, it rose to 11.921 picoamps. Furthermore, the researchers noted slower MEPSC decay kinetics.
Slower decay kinetics, meaning that every time an excitatory signal hit the neuron, the electrical charge lingered longer before dissipating.
Yes, the cell is being held in a prolonged, highly excited state.
It is the definition of profound hyperexcitability. The deep cortical layers are essentially screaming at the striatum.
They are. But here's the critical pivot of this 2023 paper. The striatum does not process this screaming equally.
What do you mean?
Chronic stress fundamentally skews the switch yard.
Oh, so it plays favorites between the D1 and D2 pathways?
It absolutely does.
Yeah.
The stress selectively and dramatically potentiates the direct pathway. The red D1 GO neurons. The excitatory drive onto the GO pathway skyrockets, while the indirect pathway, the green D2 stop neurons, remains relatively spared or unaffected.
Okay, so the operator controlling the gas pedal is suddenly receiving twice as many frantic, loud orders to accelerate, while the operator on the brakes is just sitting there at baseline, receiving normal traffic.
Exactly.
The structural bias heavily favors raw action over suppression.
Yes, but Rodriguez and Montero found something even more insidious than just hitting the gas.
It gets worse.
It gets worse. They found a biological double whammy that they explicitly term disinhibition.
Okay, walk me through disinhibition.
So the striatum isn't just populated by those medium spiny projection neurons. Woven throughout the network are these crucial localized regulatory cells. Fast-spiking parvalbumin, or PV inhibitory interneuron.
Fast-spiking PV interneurons.
Right. Think of them as the local traffic cops stationed right next to the switch operators.
Okay.
Their entire biological function is to release GABA, which is an inhibitory neurotransmitter, to suppress the projection neurons and prevent runaway, uncontrolled firing.
So they are a localized, hardwired, fail-safe mechanism right there on the floor of the switchyard.
Exactly. And what the researchers discovered is that chronic stress actually reduces the excitatory drive that normally activates these vital PV interneurons.
Oh, that is absolutely terrifying.
It is.
If we map that back to our train yard, the stress slams a cinder block onto the gas pedal of the D1 GO pathway, forcing it into massive overdrive. And simultaneously, it takes a pair of wire cutters and severs the local brake lines, the PV interneurons, that could have stopped it locally.
That is precisely what happens. The net physiological result is total, unchecked strival disinhibition.
Wow.
The direct pathway is running rampant, completely untethered from both top-down PFC control and local interneuron regulation.
See, this isn't just an abstract observation about glowing cells in mouse brains. This translates directly into deeply painful human clinical realities.
Oh, incredibly painful realities.
When your D1 GO pathway is biologically locked in overdrive and the brakes are cut, what does that actually look like in human behavior?
The authors explicitly connect this state of profound basal ganglia disinhibition to the emergence and exacerbation of severe behavioral rigidities. We're talking about motor stereotypies, uncontrollable compulsions, and encheritic disorders.
So this is the mechanism.
It provides a direct electrophysiological explanation for why conditions like Tourette's syndrome or obsessive-compulsive disorder are so famously hypersensitive to stress.
Right. If someone with OCD is managing their symptoms well on a good day, their PLC is likely exerting strong top-down control over the switch yard, bolstering the stop pathway.
Modulating the balance.
But the moment they undergo chronic stress, say they go through a horrible divorce or a job loss, the PFC shuts down, the D1 pathway hyperexcites, and those PV interneurons fail.
The brakes fail.
So the compulsion to wash their hands repeatedly or check the locks 20 times is no longer just a psychological urge. It is a literal freight train of D1 excitation that physically lacks the biological brakes to be stopped.
It reframes compulsive habits entirely, doesn't it?
It really does. They are not a failure of willpower.
No, willpower is a function of the PFC, which has been biochemically evicted from the system. The habit is just the raw, uninhibited output of a structurally remodeled basal ganglia.
This gives us such a vital piece of the puzzle. We know how the switch yard breaks, but, and I keep coming back to this, it leaves a glaring, almost uncomfortable mystery right in the center of the narrative.
What's that?
The basal ganglia is an execution center. It routes the trains, but it doesn't build the tracks.
Sure.
If the PFC is offline, who is actually stepping up to the microphone and ordering the basal ganglia to form these new, rigid habits, where is the master signal coming from?
To find the architect of the habit, we have to arrive at the very cutting edge of modern neuroscience.
The 2025 paper.
Yes. We are looking at a landmark paper published just recently, in 2025, in the journal Nature. The research was led by Jacqueline R. Giovaniello alongside a massive team of neuroscientists.
And the title.
The paper is titled, A Dual Pathway Architecture for Stress to Disrupt Agency and Promote Habit.
Disrupt agency and promote habit. That is a bold title for a Nature paper.
It is, and the methodology they use to map these deep brain circuits during active learning is just staggering.
Because they didn't just record the electrical chatter like the 2023 paper, right?
No, they actively intervened in it. They utilized a highly sophisticated systems neuroscience approach, deploying optogenetics in awake, behaving mice.
Optogenetics. Let's expand on how profound this technology is for a second.
It's basically science fiction brought to life.
It really is. In this study, they used a protein called channel rhodopsin. And when you implant microscopic fiber optic cables directly into the mouse's brain and pulse a specific blue laser light, any neuron expressing channel rhodopsin will instantly fire.
Instantaneously.
You can turn a specific pathway on at the speed of light.
And conversely, they use proteins like archaeodopsin, or ARCH, which silence the neuron when exposed to green or yellow light.
So you can turn the circuit off. They are literally playing the brain circuits like a keyboard.
It gives unprecedented causal control. And Giovaniello's team used this technology to interrogate the anatomical connections between the amygdala, which we know from Arnston and Diamond is supercharged by stress, and the dorsomedial striatum, the DMS, which we know is the habit switch yard.
So how does the amygdala talk to this switch yard?
Well, they meticulously mapped two distinct parallel pathways, projecting from different nuclei of the amygdala directly into the striatum.
Pathway 1 and Pathway 2. Let's break down this architecture of agency versus habit.
Okay, pathway 1 is the BLA to DMS pathway.
BLA to DMS.
That is the projection from the basolateral amygdala down to the dorsomedial striatum.
Got it. And Pathway 2.
Pathway 2 is the CCA to DMS pathway. That originates in the central amygdala and also projects to the dorsomedial striatum.
Okay, so two different regions of the fear center projecting into the exact same switch yard. Under normal, healthy, unstressed conditions, what is the functional difference between these two pathways?
The difference is night and day. The basolateral amygdala, the BLA, is highly sophisticated. It doesn't just process raw fear, it is crucial for learning the specific value of outcomes.
By you learning.
Right. The researchers used optogenetics to monitor and manipulate these pathways while mice learned a task for a food reward. And they discovered that the BLA to DMS pathway is fundamentally the pathway of agency.
Agency meaning flexible, adaptable control.
Exactly. The BLA to DMS pathway supports action outcome learning. It is actively recruited when an animal is analyzing the consequences of its behavior.
Give me an example of what that looks like for the mouse.
It allows the brain to understand, if I press this specific lever, I get this specific reward. But wait, the environment just changed. The reward is no longer there. Therefore, I must flexibly alter my action and try a different lever.
So it requires constant active updating of the world's parameters.
It does. It's intelligent adaptability. It's analyzing the return on investment for every action.
That's the BLA. What about the other pathway, the central amygdala to the striatum?
The CA to DMS pathway operates on entirely different logic.
How so?
The researchers proved that this pathway explicitly and uniquely mediates the formation of rigid, routine habits.
So it's the anti-agency pathway.
Essentially, yes. The central amygdala does not care about the changing value of the reward. It does not update its models based on environmental feedback.
What does it do?
Its only imperative is the relentless repetition of a previously reinforced action. Just keep pressing the lever. The context doesn't matter. The reward doesn't matter. Just repeat the action.
Okay, I want to expand our analogy of the factory floor to really cement this dynamic. Imagine the dorsomedial striatum. The switchyard we've been talking about is a massive manufacturing plant.
Okay.
The basolateral amygdala, the BLA, is the innovative, highly adaptable floor manager.
The good manager.
Right. This manager walks the assembly line, watches the market trends, listens to customer feedback, and says, look, the demand for this specific part just dropped. Stop the machines, let's retool, and start building something more valuable.
That is flexible action outcome learning. That is agency.
It is responsive to the environment. But the central amygdala, the CEA, is a strict, old-school, authoritarian foreman.
The bad boss.
The worst boss. This boss never looks at the market, ignores all feedback, and simply screams at the workers, I don't care if the product is useless now. We have always stamped the metal this way, so keep stamping it exactly the same way over and over, faster.
That is rigid habit formation.
That analogy perfectly captures the tension between the two circuits, doesn't it?
It really does. Under baseline conditions, there is a dynamic interplay between the two managers. But the crux of the 2025 paper, the absolute breakthrough, is what happens when you introduce chronic stress into this delicate architectural balance.
All right, because we know stress breaks things. They took mice, subjected them to chronic stress, and then tried to teach them a new task. What did the lasers reveal about the factory managers?
The lasers revealed a complete and devastating biological hostile takeover.
A hostile takeover.
A history of chronic stress actively and profoundly abolishes the reward-evoked activity in the BLA to DMS pathway.
Abolishes it.
The stress literally silences the innovative manager. The signal is cut. The brain entirely loses its neurobiological capacity to learn flexible action outcome contingencies.
The biological mechanism for agency is effectively uncoupled from the execution center. The innovative manager is locked out of the factory.
Yes, they are gone. But the striatum still needs instructions. Nature abhors a vacuum.
Right, the factory has to keep running.
Exactly. So while the BLA agency pathway is aggressively suppressed, chronic stress simultaneously causes the brain to progressively and powerfully recruit the CA to DMS pathway during the learning process.
The authoritarian foreman is handed the master key and the megaphone.
The stress actively promotes the premature, accelerated formation of inflexible habits.
It structurally forces the brain to abandon flexibility.
It shifts its reliance entirely onto the central amygdala circuit. The brain learns the new environment not as a puzzle to be solved adaptively, but as a rigid routine to be blindly repeated.
And when you layer this 2025 discovery over the 2023 patch clamp data we just discussed, the true terror of this mechanism snaps into focus.
Really does.
Because the central amygdala is screaming, habit, repeat, down into a striatum where the D1 go gas pedal is already stuck to the floor and the PV interneuron brakes have already been cut.
The habit is formed rapidly, it is executed with massive excitatory force, and it is nearly impossible to biologically suppress. It is a perfect neurobiological storm for the destruction of behavioral flexibility.
This synthesis is exactly why we do these death dives.
It's beautiful, scary science.
It is. We have journeyed through 2 decades of intensive science here. We started with Arnsten showing us that the delicious cycle of the prefrontal cortex breaks down under catecholamine flooding.
Right.
We looked at Diamond showing us how the amygdala takes over to hyper-focus on threats. We descended into Stocko's basal ganglia switchyard and saw through Rodriguez's patch clamps that chronic stress hyperexcites the direct go pathway and creates total disinhibition.
The cut breaks.
And now, Giovanniello has proven that the amygdala literally silences your agency pathway and hardwires a habit pathway into that runaway switchyard. The biology is crystal clear.
Yeah, ambiguous.
But what does this actually mean for the listener? How do we take this incredibly dense structural map and apply it to the reality of human existence?
Well, the most immediate implication is a radical shift in how we view behavioral failure. It completely strips away the moral failing, the deep shame associated with falling into bad habits under stress.
The shame is almost heavier than the habit itself. for a lot of people.
It really is.
People think, why am I so weak? Why can't I just choose to be better?
But this literature proves that in moments of chronic stress, you are not operating with the same brain you have on a calm Sunday morning.
You literally aren't.
Complex, top-down, PFC-led, BLA-mediated decision-making requires massive, sustained metabolic resources. It is energetically expensive to be a conscious, adaptable agent in the world.
Takes calories.
Yes. And when your amygdala detects chronic threat, it executes a brutal evolutionary energy-saving protocol. It shuts down the expensive cortex, blocks the agency pathway, and fires up the automated low-energy habit loop.
From a purely evolutionary standpoint, this makes perfect sense.
Total sense.
If you are a hunter-gatherer being stalked by a rival tribe or a predator day after day, you do not want to sit around flexibly pondering the nuanced variables of your escape route.
Should I go left or right? Let me analyze the terrain. No.
You don't want to innovate. You want to fall back on the most reliable, deeply hardwired automatic escape sequence you possess. Hesitation is death. Rigid habit is survival.
Exactly. The shift to habit is fundamentally an adaptive survival mechanism. But the tragedy, as Arnsten noted back in 2009, is the timeline of evolution versus the reality of modern society.
We evolved for the savannah, not the office.
Right. In the information age, physical survival against predators is rare. Survival now requires complex, PFC-mediated cognitive abilities.
We need our working memory to navigate nuanced social dynamics, complex software, financial planning, and abstract relationships.
Our modern predators are emails, mortgages, and systemic uncertainties. And you cannot fight a mortgage with a disinhibited D1 motor pathway.
As much as we might want to punch the mortgage.
Exactly. When this ancient survival mechanism triggers in the modern world, it becomes a severe, sometimes fatal liability. It drives us toward coping mechanisms that offer immediate, rigid relief but long-term destruction.
And this framework is essential for understanding clinical vulnerabilities, which brings us to several vital supporting papers in our stack.
Yes, let's look at those.
Consider A.R. Otto's 2013 paper published in the Proceedings of the National Academy of Sciences.
A great paper.
Otto and his colleagues investigated individual variability in this stress response. They found that individuals with inherently higher baseline working memory capacity can actually protect their model-based flexible learning systems from stress.
So having a highly conditioned, robust prefrontal cortex acts almost like a biological shock absorber.
A buffer.
It buffers the system, delaying the amygdala's ability to hijack the switchyard.
Yes, it suggests that agency isn't just a fixed state. It is a capacity that can be fortified or depleted.
But on the darker end of that spectrum, we looked at researchers like Taylor in 2015, and, importantly, Lipton's 2019 paper, Dorsal Striatal Circuits for Habits, Compulsions, and Addictions.
Addiction.
Yes, these papers directly link this exact cortico-basal ganglia restructuring to the devastating cycle of addiction.
Addiction is, in many structural ways, the ultimate pathological hijacking of the habit system, isn't it?
It is the extreme endpoint of the CEA to DMS pathway. And Lipton's work, integrated with everything we've discussed today, explains one of the most frustrating and tragic clinical realities of addiction: why chronic stress is the absolute #1 trigger for substance relapse, even after years of sobriety.
Let's walk through that mechanism, because I think it removes the stigma and replaces it with sheer biology.
Let's do it.
Imagine a person who has been completely sober for 5 years. They have spent years engaging their prefrontal cortex, using therapy and conscious effort to build new, healthy pathways. Their BLA agency manager is back in charge, directing traffic away from the substance.
See how I'm managing it.
But then a massive chronic stressor hits. Their spouse asks for a divorce or they face bankruptcy. What happens in the switchyard?
The sequence we mapped today initiates instantly. The massive stressor floods the PFC with catecholamines, completely short-circuiting the executive control center that was maintaining the sobriety.
The PFC goes dark.
The BLA agency pathway is actively silenced by the amygdala.
The innovative manager is locked out.
The CA authoritarian manager takes over, looking for the most deeply ingrained, neurologically entrenched habit loop available to soothe the highly stressed system.
And simultaneously,
the D1 GO pathway becomes radically hyperexcited and stripped of its PV interneuron brakes.
so the brain defaults to the strongest groove ever carved into that switchyard, seeking the substance.
Yes.
The relapse is no longer a conscious choice made by a flawed character. It is a raw, disinhibited motor reflex executing a command from a terrified amygdala. The biology demands the habit.
Understanding this doesn't remove personal responsibility,
Yeah.
but it completely changes the therapeutic approach.
How so?
You cannot simply tell an addicted patient under stress to make better choices, because the biological hardware required to make flexible choices is temporarily offline.
You can't ask a melted engine to run smoothly.
Exactly. You have to
Yeah.
address the structural hyperexcitability and the stress load before agency can even exist again.
It is a sober, heavy realization. We are biological machines, profoundly vulnerable to the environments we inhabit. Chronic stress isn't just a fleeting state of mind, it is a physical, relentless architect, actively tearing up the floorboards of our decision-making and pouring concrete over our habits. Let's summarize the immense ground we have covered.
We began with Arnstein's mapping of the prefrontal cortex, observing how the optimal delicious cycle of neuromodulators devolves into a vicious cycle under stress, functionally shutting down executive control.
Then we integrated Diamond's temporal dynamics model, revealing that stress doesn't just impair the brain. It actively shifts resources to the amygdala to prioritize rigid, flashbulb memory formation over complex cognition.
We then descended into the basal ganglia with Stocko, mapping the conditional routing system of the D1 direct go pathway and the D2 indirect stop pathway.
We looked through the patch clamp microscopes with Rodriguez in 2023 to witness the terrifying reality of disinhibition, how chronic stress hyperexcites the D1 gas pedal and literally cuts the PV interneuron brakes.
And finally, we closed the loop with Giovanniello's 2025 Nature paper, using optogenetics to prove that stress actively silences the basolateral amygdala's agency pathway, while powerfully recruiting the central amygdala to hardwire rigid habits into that disinhibited striatum.
It is a perfect, flawless mechanism for surviving a tiger attack, and a perfect mechanism for destroying your life in a modern office.
Well said.
But I don't want to end this deep dive on a note of biological determinism. I want to leave you, listening to this, with a final provocative thought, a hypothesis to chew on as you navigate the stress of your own life.
Science is fundamentally about finding the levers of change.
Exactly. We now know, with microscopic optogenetic certainty, that chronic stress physically rewires the amygdala striatal circuits to prioritize rigid habits. It silences the basolateral amygdala and amplifies the central amygdala, but neuroplasticity is a two-way street.
It absolutely is.
If the brain can wire itself into a cage, it must possess the mechanisms to wire itself out.
The architecture is dynamic, not static.
So the lingering question is this. Could the reverse of this process be deliberately engineered?
That is the big question.
Can we actively, intentionally engage in practices, whether that is deep mindfulness training, targeted working memory expansion like auto-suggested, or throwing ourselves into deliberate, highly focused, novel learning environments that physically strengthen that BLA agency pathway?
Could we build a buffer?
Right. Could we, through deliberate, conscious effort when we are not stressed, effectively build a biological shield, a reinforced architecture of agency that is robust enough to protect our prefrontal cortex from collapsing when the inevitable, unavoidable stresses of modern life finally hit?
That is the frontier. If neuroplasticity can lock us into an automated habit, directing that same plasticity must hold the key to permanently reclaiming our free will.
Thank you for joining us on this incredibly deep, complex journey into the architecture of your own behavior. Protect your prefrontal cortex, guard your agency, and we will see you next time.